Hello everyone, welcome to the course of Microbiology. The course code is MBT-202 for MSc Biotechnology second semester students. Today's topic is Methods of Microbial Growth Estimation. So, there are many ways to measure microbial growth to determine growth rate constant and generation time. However, the most appropriate approach depends on the experimental situation. So basically, the microbial growth estimation methods can be explained in two ways. One is the direct methods, and another methods they are comes in the category of indirect methods. So actually, direct method itself it is further of two types. One is direct measurements of cell numbers, another is viable counting methods. In direct measurement of cell numbers, the total cell count, the total number of cells are counted. It means it includes dead as well as live cells. While in case of viable counting methods, only the cells which are able to divide, they are counted. So direct measurement of cell numbers, it can be done either Petrov counting chamber method, which actually utilizes a microscope, light microscope, then Coulter counter method, which utilizes an electronic device, and flow cytometry, which is the advanced one and most popular nowadays. While viable counting method, it depends upon the plate count techniques. Plate count techniques can be of four types. Four plate method, that is very classical, traditional one. Four plate method, split plate method, and straight plate. Another method which utilizes the membrane filter, it is known as membrane filter method. And generally, it is used for the counting the microbial counting in a water sample. Then another method which is known as most probable number, it is also in use to count the approximate number in a particular sample, environmental sample. This is all about the direct counting methods. While indirect counting method, it depends upon the either measurement of cell mass, total cell mass, and measurement of cell mass. It itself can be done by measuring the dry weight or by observing the turbidity. Another method, indirect method, is the measurement of metabolic activity. The metabolites which are actually produced or as the cell divide, the number of cells increases, the total mass of the particular metabolites, for example, protein, it also increases as the uh, cell number increases. So let's talk about the direct method of the microbial growth estimation. So generally, first of all, we would like to talk about the flow cytometry method. And this flow cytometry method, it is actually uh, uh, it is increasingly being used to directly count microbes and to get the detailed information about the microorganisms. So, flow cytometry method, it is the principle of flow cytometry. You can see in this figure. And a flow cytometer actually creates a stream of cells so narrow that one cell at a time, you can see here, it can pass through uh, the beam, the laser beam. It can pass through a beam of laser light. As each cell passes through the beam, the light is scattered. As uh, when the cell it is passed through this laser light source, so what happens? The light it is scattered by the cell, and scattered light is detected by the flow cytometer. Here it is a detector which actually detect the fluorescence emitted from stem cell. Actually, in case of flow cytometry, the cells are labeled with some fluorescent dye. So as soon as this light is scattered, the detector it detects the fluorescence emitted from stem cells, and the number of actually as we know that the cells are separated in space because there is no enough space to pass more than one cell, so only one cell can pass through this space, and each light scattering event is detected independently. So because one cell it is responsible for producing a particular signal at a time. So the number of light scattering events it represents the number of cells in the sample. So and not only the number of cells of different size, internal complexity, and other characteristics within a population can also be counted with the help of flow cytometer. So it is the advanced technique to directly count the cell number, and this technique is very popular nowadays, and uh, generally for counting not if we are studying the different properties of cells like size, internal complexity, and other characteristics, for that purpose usually use of fluorescent dye or fluorescently labeled antibodies are used. So actually there are more sophisticated flow cytometers are available, which can provide valuable information about characteristic of of the population of cells. Next method is the Coulter counter method. Actually, a Coulter counter it is an electronic device, and in the Coulter counter, you can see here. This is the picture of Coulter counter, and it possesses a very small orifice here. This orifice is present over here, and through this orifice only a single cell can pass, and this Coulter counter it possesses two electrodes, positive and negative electrodes, and when the voltage is applied the current to flow through this uh, electrode, and when a cell it is passed through this hole, it actually resists the flow of the current, and this resistance can be measured, and it is actually the direct measurement of the uh, cell number. So, in the other word, you can say that in the Coulter counter, a microbial suspension is actually deliberately forced through a small orifice, and electrical current which flows through the hole and electrode placed on both sides of this orifice It measures electrical resistance, and every time when the cell it passes through this orifice or hole, it might the electrical resistance increases, or you can say the conductivity drops, because conductivity is inversely proportional to the resistance, and by this way the cell is counted. It is a traditional method for directly counting microbes uh, in a sample, but the drawback of this Holter counter method is that it counts both dead and live cells. So you cannot actually count the viable cells from this by this equipment. However, with the uh, use of this flow cytometry or flow cytometer, if we prepare fluorescent dye that can differentiate between live and dead cells, it is possible to count directly the number of live and dead microorganisms in a sample. So, the flow cytometry method is much more valuable in comparison to the Holter counter method. Now we move towards the Petrov counting chamber method, which is actually very popular method to count the cells. Particularly, it is the cytometer, hemocytometer, which is a type of Petrov counting chamber method. It is very popular for estimating the total mm, leukocyte count and differential leukocyte count. So, in case of Petrov counter, which is actually it is a specialized type of slide. This counting chamber actually it consists of a specially designed slide and cover slip, and the space between the slide and cover slip, it you can see here the space between this is the slide, this is cover slip, and the space between this slide and cover slip, it creates a chamber of known depth, and the depth of this chamber is 0.02 mm. Depth is 0.02 mm, and here the chamber is this chamber, it actually contains 25 squares. Here you can see 1 2 3 4 5, and similarly in 5 lanes. So total number of chamber is 25, and this 25 squares it covers an area of 1 mm square. So it means this length and width, they are of 1 mm in measurement. So the area of 25 square it covers the 1 mm square. So the total number of bacteria in 1 mm square of the chamber, it can be calculated the number of bacteria in 1 square multiplied by 25. Okay. So actually this chamber, it is not only 1 mm by length or 1 mm by width, but it is also 0.02 mm deep. So if we count the number of microorganism or number of bacteria in a particular volume of this chamber, so it can be represent as you can see here the number of bacteria per square multiplied by 25 multiplied by 1 upon 0.02. which is equivalent to the 50. Is it clear? So number of bacteria per cube, it is equal to number of bacteria. For example, if we count mm, 25 number, here it is given as an example, 25 number of bacteria in a square, in a small square, a gridded square. So total number of bacteria which is present in 1 cube, it could be calculated as 25. That is the number of bacteria per chamber into 25 squares into 50, which comes when we put the value of the depth, that is 0.02, 1 upon 0.02, that is equal to 50. So total count which comes, it is approximately 3.125 into 10 to the power 3. And if we want to calculate the number of bacteria per centimeter cube, it could be 10 to the power 3 times of this value. So when we calculate the average count per square in 25, if we want to count the total number of bacteria in this Petrov household counter, and the average count per square is 25 bacteria, then the value will come 3.125 into 10 to the power 7. Is it clear to all of you? So one disadvantage of using this counting chamber is that to determine population size accurately, the microbial population, uh, it must be relatively large and evenly dispersed, because only a small volume of the population is sampled here. केवल एक छोटा सा वॉल्यूम जो है in microliters, it is used as a sample when we from a huge volume of the sample. So it is not very accurate counting method. So next methods of microbial growth estimation, indirect method, is the counting the viable count, that is plate count techniques. So in plate count techniques, first of all, if we want to count the number of bacteria or microorganisms from a environmental sample, it is necessary to obtain a pure culture first. Okay. किसी भी सैंपल में एक पर्टिकुलर पॉपुलेशन को स्टडी करने के लिए कि उस पॉपुलेशन का पर्टिकुलर एनवायरनमेंट में, पर्टिकुलर नेचुरल हैबिटेट में क्या उसका जो है ओवरऑल टोटल नंबर है, और इफ वी वांट टू ग्रो इट इन ए लेबोरेटरी कंडीशन, पहले तो आपने नेचुरल कंडीशन में देखना चाहा कि हाँ कितना नंबर ऑफ काउंट है, तो यू कैन यूज एनी ऑफ द डायरेक्ट मेथड। और उसके बाद आप अगर उसका जनरेशन टाइम निकालना चाहते हैं या ग्रोथ रेट काउंट करना चाहते हैं और उसकी अदर बायोकेमिकल और मॉलिक्यूलर कैरेक्टरिस्टिक स्टडी करना चाहते हैं, तो यू मस्ट फॉलो द इट्स रिप्रोडक्शन बिहेवियर, और उस रिप्रोडक्शन बिहेवियर के लिए यू मस्ट गो फॉर द सेल काउंट। तो सबसे पहले या जनरेशन टाइम जो है आपको देखना पड़ेगा, जनरेशन टाइम क्या है कि कितने टाइम में वो जो है बैक्टीरिया डिवाइड करता है, डबल हो जाती है पॉपुलेशन। तो उसके लिए आपको सबसे पहले प्योर कल्चर आइसोलेशन करना पड़ेगा। और प्योर कल्चर आइसोलेशन के दो मेन मेथड हैं। वन इज सीरियल डाइल्यूशन फॉलोड बाय आइदर पोर प्लेटिंग और स्प्रेड प्लेटिंग, और अनदर मेथड इज द सीरियल डाइल्यूशन फॉलोड बाय स्ट्रीकिंग। और प्योर कल्चर कैसे होगा? जब आप जो भी आप सैंपल ले रहे हैं और उसको सॉलिड मीडिया पे ग्रो कराएंगे, क्योंकि लिक्विड मीडिया से आपको हमेशा, अगर आपने डायरेक्ट सैंपल जो है एनवायरनमेंट से कलेक्ट किया है, तो आपको हमेशा मिक्स कल्चर मिलेगा। इन नंबर ऑफ माइक्रो ऑर्गेनिज्म आर देयर, ठीक है, दे बिलोंग्स टू डिफरेंट स्पीशीज, जीनस एंड इवन different family or even different kingdom. So आप को चाहिए प्योर कल्चर। तो प्योर कल्चर के लिए यू मस्ट हैव सिंगल डिस्क्रीट कॉलोनीज। ये जो आपको डॉट्स दिख रहे हैं, दे रिप्रेजेंट सिंगल कॉलोनीज, और दूसरी कॉलोनी इसके आसपास इसको कंटामिनेट करते हुए नहीं होनी चाहिए, ना ही कोई ओवरलैपिंग कॉलोनी होनी चाहिए। This colony must be discrete and grow as a single colony. तो इस सिंगल कॉलोनी को ऑब्टेन करने के लिए we have two important method. One is the serial dilution followed by core plating or spread plating method, and another is serial dilution followed by स्ट्रीकिंग मेथड। तो स्ट्रीकिंग में भी आपको यहाँ पे एक प्रॉपर टेक्निक होती है टू ऑब्टेन द डिस्क्रीट कॉलोनी। हियर यू कैन सी, जहाँ पे कर्सर है, कि आपको सिंगल डिस्क्रीट कॉलोनी दूर दूर दिखाई पड़ रही है। अदरवाइज शुरुआत में जो यहाँ पे स्ट्रीकिंग जहाँ से शुरू की गई है, वहां पे आपको ओवरलैपिंग कॉलोनीज है, यू आर अनेबल टू आइसोलेट ए सिंगल कॉलोनी। So you have to obtain a discrete and single colony, और उससे फिर आप प्योर कल्चर आइसोलेट करते हैं। आफ्टर obtaining the pure culture, and what is the serial dilution? Actually, if you have an original sample, for example, you have an original sample, so you have to dilute it by many times. So for that purpose, you have to take 9 ml of sterile water, or whatever a constant volume you should take, and add, for example, if you take 9 ml of, ये ज़्यादा आसान है समझना, 9 ml of sterile water and add the 1 ml of original sample, it means total volume in this tube is 10 ml. ओके, सो 10 ml में जो आपका ओरिजिनल सैंपल है वो कितना है? 1 ml. तो दिस 1 ml ओरिजिनल सैंपल, इट इज डाइल्यूटेड बाय 10 times. So this is considered as 10 to the power minus 1 dilution. This is considered as 10 to the power minus 1 dilution. Then after this, another tube has been prepared which contains another 9 ml of sterile water, और जो ये 10 to the power minus 1 diluted आपका सैंपल बना है, उससे 1 ml लिया और इस 9 ml sterile water में ऐड कर दिया इनोकुलम। दिस डाइल्यूशन इज अगेंस्ट 10 times, और 10 into 10 कितना हुआ? 100 टाइम डाइल्यूशन हो गया। सो इट इज रिप्रेजेंटेड एज 10 to the power minus 2 dilution, and so on. So after diluting the sample, डाइल्यूट क्यों किया जाता है? क्योंकि आपका ओरिजिनल सैंपल में इट इज अज्यूम्ड कि इट कंटेन्स नंबर ऑफ माइक्रोब्स। तो आपको क्या करना है कि जो नंबर, अगर इनको आप एज सच किसी प्लेट पे कर देंगे तो ये ओवरलैप कर जाएंगे, लेकिन अगर आप इनको जो है डाइल्यूट कर देंगे, तो मान लीजिए आइडियली जो है 10 to 150 microbes should be present in your inoculum, जिससे कि आपको 10 से लेके और 150, जो आइडियल कंडीशन है 150 बहुत अच्छा दिखता है। इफ यू हैव टेकन दिस 90 mm पेट्री प्लेट, तो इसमें आपको डिस्क्रीट कॉलोनीज मिल जाएंगी। तो एक अगर 10 या से लेके 150 तक नंबर ऑफ वायबल सेल्स प्रेजेंट है, वी आर टॉकिंग अबाउट द viable count. So viable cells are those cells which are able to reproduce. They are live and able to reproduce. Okay, so these viable cells, if they are present in a concentration of, say for example, 25 to 150 number, so, and if they are spread, a spreading method है, in which we take an L shape. You can see here L shape spreader. This can be of metallic or glass, and this spreader first is dipped in a 70% alcohol, and then it is sterilized through direct flaming method. After direct flaming, this spreader is cooled down so that it can use to evenly spread the cell from this serially diluted sample onto an agar medium plate. So when this L-shaped spreader is used, this method is known as spreading method. Another method to obtain the pure culture colony is the pour plate method. In pour plate method, we just put a small amount of, say for example, 100 microliter or 50 microliter of this diluted sample on agar top agar plate, and or directly on the malted agar medium, which should be of temperature near about 45 degrees Celsius, so that there is no adverse effect of temperature on the bacterial life or microbial life. So after cooling the agar medium up to the 45 degrees Celsius or 40 degrees Celsius, a small amount of diluted sample has been added into the Petri plate along with the molded agar, and the plates are filled so that the sample, it is evenly distributed into the molten agar, and then it is incubated for uh, 8 to 24 hours to obtain the discrete pure colonies. And these discrete pure colonies then counted, and this counting is known as viable counting or plate count techniques, which tells about the plate forming colony forming unit, that is known as uh, CFU, colony forming unit per ml of the diluted sample. So this is the plate count technique, which involves three methods: spread plate method, pour plate method, and streaking. Another method is the use of uh, uh, this filtration method, the membrane filtration procedure. And in this membrane filtration procedure, the membrane filter is used. This membrane filter has a pore size of 0.45 micron, and the environmental sample, that could be in liquid form, uh, and it is passes through the membrane filter on a filter support, and after that, uh, when this water sample filtered through membrane filter, the membrane filter removed this membrane filter removes, which holds in constant number of viable cells, and this membrane, it is then placed. You can see here in this figure, this membrane, which contains a number of viable cell, it is placed on a appropriate agar medium in a petri plates, and then this is allowed for incubation for 8 to 24 hours depending upon the result. अगर 8 घंटे में आपको रिजल्ट मिल जाता है then 8 hours incubation at a particular temperature or optimum temperature is sufficient, and then you can see the result in the form of typical colonies. So these colonies can be counted, and these colonies can be represented as colony forming unit per ml of the sample. So, for example, there is one example in which um, the method is used in case of plate counting method, how to determine the colony forming unit per ml, and the sample, it is actually ideal that sample should yield between 25 and 250 colonies per plate for accurate counting. And once the number of colonies is known, the original number of viable microorganisms in the sample can be calculated from the number and the sample dilution. For example, if 1 ml of solution of 1 ml of a solution diluted by a factor of 1 into 10 to the power 5, it yields 150 colonies. For example, 150 colonies is quite ideal for obtaining the pure culture, as I have told you earlier. These 150 colonies means 1.5 into 10 to the power 2 colonies. And the original sample, it because the 1 ml of sample which we have taken, which actually gives you 150 colonies, it is diluted by 1 into 10 to the power 5 times. So that number of colonies must be multiplied by the dilution factor. So it is equal to the original sample. It must contains around 1.5 into 10 to the power 2 plus 10 to the power 5. To overall 1.5 into 10 to the power 7 cells per ml. We are talking about the original sample. So the results are actually expressed in terms of colony forming units, and these colony forming units means the viable cells uh, which are able to reproduce, or they are able to produce a colony. So this is how this direct method of viable cell count can be done. Another method which is known as most probable number method or approach, it is used because sometimes plate counts cannot be used to measure population size, and one reason for this situation is that sometimes uh, plate counts are not helpful if the microbes cannot be cultured on solid medium, or if large colonies overgrow the surface of the plate, which is actually happen with the soil bacteria. So it is actually impossible to get an accurate count, and for those cases this most probable number approach is used for determination of the approximate count of the microorganisms. And in this method, numerous replicates of several dilution of a culture are made and added to a tubes containing a suitable liquid growth medium. After incubation, each tube is examined to determine if growth occurs. And it is assumed that the last tube in the dilution series that actually demonstrate the growth, it was inoculated with between 1 and 10 cells, and the next tube had between 11 and 100 cells, and so on. And if no growth is observed, then the tube is assumed not to have received any cells. And in this way, the number of cells in the original sample is estimated, and this method or this MPN values are most commonly used when a selective medium can be employed that supports the growth of a specific type of microbe. There are two specialized, however, if we are talking about the culture medium, they are broadly classified into two types. One is complex medium, another is chemically defined medium. Chemically defined medium has the component in molar concentration is known, while in complex medium, the exact concentration of a particular nutrient it is not known. So these mediums are further divided on the basis of their nature, that which medium supports which type of microbial growth. And on this basis, there are two type of medium: selective medium actually it selectively promote one type of bacterial growth, and it suppress the other type of bacterial growth. And on the basis of the nutrient abundance or a nutrient concentration, the medium can be classified as minimal medium, which is actually which has one or more nutrient as a limiting factor, and another is enriched medium, and another type of media which is known as differential media, which differentiate the two type of microorganisms on a culture medium. So particularly it is used for the uh, those bacteria which are actually utilize RBC and causes the red blood cell lysis, and to differentiate those bacteria from other which do not cause the lysis, and so on. So we are talking about the MPN. So MPN actually method, it is generally employed uh, on when a selective medium can be employed that support the growth of a specific type of microbe. So next method is the indirect method. And in indirect method, uh, either we go for the measurement of cell mass, that is techniques for measuring changes in cell mass, and these techniques are very popular and very traditional method to determine the microbial cell growth. And another approach is to determine the microbial dry weight, and in this case the cells growing in liquid medium are collected by centrifugation, washed, dried in an oven at a particular temperature and weight. And And this is actually useful technique for measuring the growth of filamentous fungi, and it is the drawback is it is time consuming and not very sensitive method. It is very rough method for calculating the overall or growth of the particular population. And another very popular method which is generally utilized in the laboratory, and that method is known as the measurement of cell mass by uh, measuring the turbidity of the cell culture. And for this purpose, a very popular method that is known as spectrophotometric method, it is used. And as we know that in case of spectrophotometric method, the as the number of cells uh, increases, the scattering of light also increases, because spectrophotometry depends on the fact that the microbial cells, they scatter light and that strikes them. So you can see here, this is the light source, and this is the tube which contains the bacterial suspension, and as the number increases, the light scattering property also increases, and as light scattering property, if it is increases, it means transmission decreases and absorbance increases. And this absorbance, it is directly proportional to the concentration of the cell. So by this way we can uh, measure the overall increase in the mass of the cell. So because microbial cell in a population are of roughly constant size, and the amount of scattering is directly proportional to the biomass of cells present and indirectly related to the cell number. So the concentration of bacteria when it is reaches about a million, about a million cells per ml, the medium appears slightly turbid, and when this suspension or culture, it is read against 600 nanometer, then it actually the light is scattered, and the amount is scattered, it can be measured by a spectrophotometer, and it is known as absorbance or optical density of the medium. And absorbance, it is linearly related to the cell concentration, and it is true for absorbance level less than about zero point 0.5. If the sample exceed this value, it must first be diluted and then absorbance measured. So the condition is that the absorbance level must be less than zero point 0.5. So this is how we can measure the cell mass of a particular cell suspension or microbial cell culture. Another method to estimate the cell mass is by measuring the concentration of some cellular substances. For example, a sample of cells can be analyzed for total protein, and an increase in the microbial population obviously will reflect a higher total protein levels. So another way In case of photosynthetic bacteria, particularly phototrophic cyanobacterial population, is to estimate the chlorophyll amount. So it directly correlates with the number of cells. So these are various methods, direct and indirect method, to observe the microbial growth. And with the help of these methods, you can calculate the growth rate as well as the generation time. And as we know that generation times play very important role for studying various biochemical as well as molecular studies about the particular microbial population. Thank you very much.